So to do this, we need a strict collaboration between the companies, and we are, academic partners, and also we would like to involve all the time regulatory authorities, because they have to understand what we are doing. And this is the strength point of a public-private partnerships. No? We work together in an open situation, controlling each other. You heard a lot about IP issues. No? We also this morning talk about IP issues, very important. But I think the system, and also the colleagues before, he said that years ago maybe it was not possible in companies to think to put available some data, and now in this context we can do, and we have 4 major companies that are working together, and we'll have to work together. So also, of course, the contest is made that will be a coordinator from the EFPIA side, a deputy coordinator, but I think nothing against if we keep all the colleagues involved, and we like to change the rules a little bit, no, particularly. So what are the major objectives? Of course, we talk about early inflammation. It's already quite clear that when you add adjuvants, now all the companies have adjuvants, and you increase a little bit of inflammatory response. But this is part of nature. And when you increase inflammation, in the vaccine field you talk about reactogenicity. But it means that it's working, and then there are data that are coming out in human already, that when you use adjuvants, the breadth, I would say, of the immune response of antibodies is larger than the usual one, and you discover epitopes that were not there anymore. So can we have to cope with this early inflammation? Is that the price that we have to pay? And also allergic responses, still not related, but since we are looking, I think also allergies, you know, 1/3 of the population is affected by allergies, so it will be very difficult to assess something, but you might have exacerbation or things like this. The difficult part is this one: identification and validation of early biomarkers of autoimmunity. Once you know this, you can decide or not some person should be vaccinated because it's preventing a potential risk, but maybe is exposing the person to another risk. So this, I think, is the central role, even though, as I say, there are no proofs that vaccine will induce autoimmunity. There is just a big fear, and this is affecting the use of vaccines. So this requires that the analysis will be done with the incidence of disease, and also to have a big data, large database of epidemiology on autoimmune disease in the general population. And also, last but not least, the genetic background is very important. And the previous events will involve also previous exposure to other infections. So with this, we have the expectation to modify the vaccine development, as we say, creating awareness in the industry and academia of the immunosafety of molecules impacting the immune system. It's not a case that vaccines are somehow considered something not very safe, but this is due more to the tradition. In the last century, when vaccines were developed, experiments were done in orphans and things like this. Now things are very controlled. Now vaccines are safe, but we have to get rid of this impression that that vaccines are somehow dangerous, and because otherwise the price is very high. You have a tool which is not very expensive, which is very effective, and you don't use it. Then, as you say, we have to establish a common language on the vaccine immunosafety problems, because sometimes it's also a problem of language, and we have to have the language having an impact on the general population as well. This might change also the way vaccine will be developed. There are a couple of issues that are becoming very important. As I say, now you have a lot of vaccines available, and babies can get 3, 4, 5, 6, 7, 8, 9 different vaccines at birth. So also the fact that you have all these tools that are made available, you have to start thinking that you have to immunize with different antigens and also maybe immunize not too often. There are things that still need to be clarified. And again, we are thinking the same thing, addressing the problem, safety of vaccine much earlier than before, not before the problem is coming. And this to us is mainly due, of course, developing, you will see, animal models, but we are believers that a good search on the human immunology, and we have the tools, will be important. The major goals will be identification of biomarkers of early inflammation, and with this is already clear. There are genes of cytokines, IL-1, IL-6. IL-1 is coming back, very important in a lot of factors. But so you have the tools to see this. Identification and validation of biomarkers of autoimmunity. This is the key part, no? So are there biomarkers of autoimmunity? Is there a space to find new one? Is there a space to find maybe one general that could speak for all the other situation? And the incidence epidemiology of early reaction and autoimmune disease in general population. And this is, I think, will be a major deliverer of the project, having this data in database and made available to everybody. Of course, and we have been discussing this before, we have been asked to already design a project, but it's clear that is only a suggestion. It's clear that that could be modified, no? According also to my people, as I say, the idea to go to academic people and is to find creativity and innovation. If we have all the creativity and innovation, maybe there is no need, but I think we really believe in this thing. So I don't think there is the reason to follow strict work packages. But this is just breaking down what we said before, biomarkers for inflammation, reliable animal models in vitro. Some people are still in favor of animal models. Some others are thinking, and now we are at the point that everything can be looked in human with the modern tools, because we don't need millions of cells of things. We're just going at the genomic level, which is becoming cheaper and cheaper and cheaper. The annotation may come the other way, but this we expect in the proposal of the public consortium. So these markers, and this is maybe related to the animal model much more, could reveal risks for chronic disorders after vaccination. And then the project is changing a little bit, because if you want to know whether the vaccines are affecting the normal situation, you have to know what's the normal pathological situation. Are these vaccines increasing the risk for autoimmunity? And so what's the proportion of autoimmunity in the general population? And to do this, I think the budget available in the 5 years of the project are not enough, of course. So we have to rely on data already existing in companies, data maybe of previous clinical trials made for several reasons. And so, of course, these databases are now available because there is much more capability at the IT level, knowledge level. The final outcome that, and this is, I think, very well welcomed by the companies, is a kind of general guidelines approved by the regulatory authorities. I've been discussing with colleagues, maybe some of you have seen a recent publication in Nature Biotechnology made essentially by FDA and other colleagues in the States, where they define 30 important early markers in drug toxicity for kidney cells. So at that point, this is a consensus of the 30 markers. And it's important, you know, when there is a consensus. You're going to pay a price for a consensus, but that will be the final outcome of this work. So what we expect from the consortium? Of course, it's expected to address all areas outlined in the call, even though, you know, there are different ways to interpret what is in the call. So it's new innovative approaches in the way to evaluate the immunosafety of vaccine are expected for these things at many levels, animal models, in silico models. So, of course, you are dealing with companies with a quite strong capability in so-called vaccinology, and academic colleagues are welcome. But what we would like to see more in the collaboration in clinical and preclinical identification of inflammation and autoimmunity. So this is something that is not so common in companies, to work on inflammation, to work on autoimmune disease. So the consortium has to be a good one. I'm involved in many other European projects. We see already this capability that is being promoted by the European Commission to put people to work together. Of course, it doesn't work 100% of the time, but this idea of putting industrial researchers with academic researchers, in the long run, it pays back and it creates new opportunity and also a new vision on the way of starting a research. And then the other outcome is this, maintain these biobanks. Also, this should be discussed also when we come to the point to the famous budget in kind, not in kind, to maintain data banks. I think this will be very important. And also we are very much interested in regulatory authorities, because otherwise we talk each other, but the product that we make, which is, as I said, important data, important documents to say the vaccine, if it's the case, because maybe we find out that there are holes in the system, maybe there is some autoimmunity that could come out during, after vaccination, but maybe it's just due to normal infection that we have every year. And also cohorts of patients suffering from autoimmune disease. This should be a way to look from other things. If you look at autoimmunity patient cohorts, maybe you come back to and you ask questions that were never been asked before about vaccination. What is provided by the company? The vaccines. But when I mean vaccines, it means also clinical trials. There are clinical trials every year, and that's very important. There is a need also of further discussion among the companies, because you have to understand how to operate in this system. But maybe there is also space to do a specific clinical trial, asking a new question. Why not? I don't want to influence the public consortium on the choice of diseases and so on, but like was said before, also in this case there are many vaccines, and the consortium will have to take decisions and stick with that. Share animal models that are really already in place. In companies, of course, there are many. Sometimes you have to vaccinate the mothers to protect the babies. Sometimes you have to use different routes. Talking before, using drugs, not only injecting, but in the vaccine field, also to give vaccine without the needle is important. In our population, it's important, particularly when you think to one of the major use of vaccine, which is for developing countries, where the needle is a problem. So, and again, the word harmonization and standardization, to speak the common language before. And then I just said, provide samples and things, and to put all this knowledgeable available, and in particular immunoassays that are already established in companies, because of course when you talk about antibody responses, this is very well established. When you talk about cellular immunity, that's a big hole, and still we don't know much. The colleague was talking about T-cell epitopes, but all the things are, but the practical use of a good immune response after a vaccine, cell-mediated, is still largely unknown. And maybe we can understand something more.